Welcome to the Good Chemistry Podcast. I'm your host, Nick Jikomis, and today I'm speaking with Dr. Bryan Roth. Bryan is a distinguished professor of pharmacology at the University of North Carolina, Chapel Hill School of Medicine, and an elected member of the National Academy of Medicine and the American Academy of Arts and Sciences. Bryan has been running his lab and doing cutting-edge research for around 30 years now, and his papers have been cited tens of thousands of times. He has been studying the molecular mechanisms of how drugs affect the brain for most of his career, and is also an avid practitioner of Zen meditation, which he's been doing daily for many years now. Bryan and I discussed a range of topics related to his scientific work, including receptors in the brain and how they work, how psychedelics and other drugs interact with those receptors. We talked about the massive research project that his lab is leading around the study of psychedelics and the development of new drugs to treat psychiatric disease. And Bryan also talked about his Zen meditation practice and the types of awareness that that practice can reveal. If you find the content in this episode interesting or helpful, please do consider liking, sharing, subscribing, downloading, all that stuff. I really do appreciate it. And with that, here's my conversation with Dr. Bryan Roth. Professor Bryan Roth, thanks for joining me.
Greetings. How are you today?
I'm good. And where are you actually? Where's home base for you?
So I'm at the University of North Carolina, Chapel Hill School of Medicine in the Department of Pharmacology.
Interesting. And what would you say your overarching research interest is for your lab?
So I would say ultimately we want to understand how psychoactive drugs work at the most fundamental level, and then to use that information to design safer and more effective medications, mainly for psychiatric indications. Since my background is I'm a psychiatrist, among other things.
Gotcha. So you're actually a psychiatrist, you're an MD, and you're also a PhD.
Right. Yeah. My PhD in biochemistry.
Okay. And do you do any clinical work, or is it all a lab research focus?
No, I don't see patients anymore. I did for many, many years, but I haven't for probably 15 years now.
Okay. How long have you been running your lab?
Since 1991. So that would be almost 30 years, right?
Oh, wow.
29 years. Yeah. A few years.
A few years.
Yeah.
And you're kind of known as the GPCR guy. So I thought maybe we would start with an overview of some basic biology to maybe give people some foundational knowledge that'll probably inform the rest of the discussion. So can you just talk a little bit about what are receptors, very high level, and what do they do for cells?
So I think everybody knows what drugs are, right?
Yeah, let's assume they do.
Okay, so drugs are typically, not always, they're typically small, relatively small chemical compounds. For those people that have a scientific background, typically these are compounds that have molecular weights less than 3,000, or I'm sorry, less than 350 Daltons. So these are relatively small compounds. And when you take a drug, let's take I don't know, since we're going to talk about psychedelics, let's take LSD as an example. Okay. So when people take LSD, so this is a drug, it's a small compound, to mediate its effects in the body, it has to first interact with a protein that's called a receptor. And receptors are proteins that are in cells. The proteins that the cells that we're going to talk about are cells in the brain that are important for all brain activities. These are called neurons. And receptors are just basically proteins that sit on the surface of the cell. They have openings in them for drugs to come in, drugs and neurotransmitters. And when a drug like LSD or when the natural neurotransmitter, which would be serotonin, gets in, finds its way inside the receptor, there's typically a sort of a pocket in the protein. It stabilizes a particular state of the receptor, which is active. And then once this active state has been achieved, then the receptor and the drug complex then turn on a bunch of biochemical reactions inside the cell. And G protein coupled receptors are just one sort of class of receptors that are in the body. They're the largest class of receptors that are encoded by the human genome. There are about 800 of them, between 8 and 900 of them. They're responsible from everything from vision, your sense of smell, your sense of taste, virtually all of your brain functions are mediated in part by interaction of neurotransmitters with G protein coupled receptors, sensations of pain and pleasure, and so on. So these are really, really important family of proteins in the body.
So these GPCRs, they're a certain type of receptor,
right?
And they do lots of stuff. They come in many forms, and are they all throughout the brain? Are they in certain cells, or are they everywhere?
Every cell in the body has, we'll just call them GPCRs. They're technically G protein coupled receptors, but every cell in the body has them. And every cell in the body expresses, typically 100 or so of them. So they're everywhere, and they're important for literally every everything that goes on in your body is regulated to some degree or another by GPCR. So they're really, really important.
And are these receptors, are they something that medications that a doctor would prescribe are often interacting with?
Yeah, about 30% or a third of prescribed medications have their main action by interacting with GPCRs. This includes most psychiatric medications. And then a large percentage of medications, the other 2/3 interact with GPCRs sort of secondarily. So side effects of medications may be mediated by sort of chance interaction with GPCRs. So the GPCRs may not be the main target for the drug, but the drug interacts with GPCRs, and that can be important for serious side effects.
Wow. So
they're really important.
Yeah. So every single cell in the entire body has some kind of GPCR, probably many kinds. So they come in many different flavors. So if we stick with the brain, we're talking about neurons, brain cells inside of our heads. There are all kinds of receptors that respond to many different neurotransmitters, things like serotonin and other things. We're probably going to end up talking about serotonin a lot. So what is the psychedelic receptor, or the one that's commonly associated with the effects of psychedelics?
Yeah, so that's a receptor near and dear to my heart that I've been studying since 1983. Okay, so probably I've been studying it for more years than many of the people that will be listening to this have been alive. So it's a receptor called the 5-HT2A. We can just call it the 2A receptor. And that's, there's the best evidence is that psychedelic drugs exert their psychedelic effects through activating that particular receptor. I think that's fairly well established, and probably, the vast majority of experts would agree that the 2A receptor is the target of psychedelic drugs.
So the classical psychedelics, as I understand it, are pretty much defined by their ability to bind to this receptor. So something like LSD, something like psilocybin, something like
to activate it.
To activate it.
Yeah.
Something like DMT as well. And yet they can have very different effects. Some of them last for many hours. Some of them last for just a few minutes. If this is the quote unquote psychedelic receptor, what's accounting for the differences in the effects of different psychedelic drugs?
Yeah. So that's, I would say that's something that's really unclear. So let's just take LSD and psilocybin as two examples. So psilocybin is, the drug psilocybin is actually inactive, and in your body. So when people take psilocybin, it's metabolically transformed into a compound called psilocin. So psilocybin itself is inactive. It has to be metabolically transformed to psilocin. Technically, what happens is there's a phosphate group that's removed. And it becomes a free hydroxyl, and then it's able to interact with really many serotonin receptors. And it turns out that the drugs like LSD, DMT, psilocin, I'll call it psilocybin, but just for the science nerds that may be listening to this, when I say psilocybin, just bear with me and repeat to yourself psilocin.
All right. And this is the active compound in magic mushrooms.
This is the active compound in magic mushrooms, right? So, what we have found over the years is these compounds actually interact with dozens of different receptors. And so they have this very complex pharmacology. But when there are now really well-controlled human trials where humans have been given drugs. The drug they're typically given is a drug called ketanserin, which it's not entirely specific for the 2A receptor, but it's reasonably selective for the 2A receptor. So when humans are given that drug, and it basically blocks the activity of that drug. And then when they're given, a hefty dose of LSD or psilocybin, they don't have a psychedelic experience at all. So that's probably the most compelling evidence we have that the psychedelic effects are mediated by the 2A receptor. We don't have data like that for a drug like DMT. And I suspect at some time in the future, somebody will, do a clinical study and determine if the psychedelic effects of DMT also are blocked by a 2A antagonist.
Would your intuition there be that maybe they're partially blocked, but not completely?
So I don't know. We know that in mice, for what it's worth, you can block the effects of DMT by blocking the 2A receptor. We can block many of the behavioral effect, the effects that we can measure in a mouse. Many of them are blocked by 2A antagonists for, all of the psychedelics, but not all. So there are sort of classical mouse behavioral markers of psychedelic drug action that, and technically these are things like the head twitch response, nose poking, retrograde walking, disruption of prepulse inhibition. There are, a few of these mouse behaviors that are completely blocked when mice are given highly selective 5-HT2A antagonists, irrespective of what the psychedelic drug is. So it could be LSD, psilocybin, mescaline, DMT, 5-methoxy-DMT, 25-cyano-NBO, the NBOs.
What are those? I don't know what the NBOs are.
Those are designer psychedelics. They're apparently fairly popular in Europe. I don't know if they ever made it on the market here, on the black market here. So we have, really, really strong data in rodents, and for all psychedelic drugs, and then in humans we have strong data for two, and we just don't have any data for the rest of them. Now, the other thing that is sort of that distinguishes drugs like LSD, psilocybin, and DMT. Those are sort of three drugs that in humans, if you go to Arrowwood or read data from human studies where these drugs have been given, the effects are quite different.
Mm-hmm.
And we don't know if that's because of the other receptors that these drugs hit, or if it's because of something that's called pharmacokinetics. So a drug like LSD actually takes a long time to interact with the receptor. Once it gets on the receptor, it's on there for a long period of time. A drug like psilocybin is sort of intermediate, and a drug like DMT is very rapid. It gets on the receptor very quickly,
mm-hmm.
falls off the receptor fast, and it's removed from the body very quickly. And typically when people take DMT, they're given very large doses. And one can imagine that the receptors are basically fully occupied nearly instantaneously by DMT. Where that's never going to happen with a drug like LSD. LSD takes actually a long time to get on the receptor, and psilocin is sort of intermediate. So it could in part be due to what we call pharmacokinetic things, or it just could be that a drug like DMT, which is sort of a real outlier, has other pharmacology that's important for its action. And as I said, these drugs interact with dozens of receptors, so it's conceivable that something else is going on. It's also possible that they interact with the receptor differently. So, but we basically need definitive data. We just don't have it. So.
Gotcha. So some of these drugs are literally getting onto the receptor very slowly in the case of LSD, and
very slowly.
And it takes a long time. So they might get on some receptors, but not all of them. And then they get on some more later. And then by then, maybe some of it's washed out or something, where something like DMT, it sort of all hops on all of the receptors it can, and then it gets right off.
Yeah. And it's removed very quickly from the body as well. So it has, it basically is degraded very quickly.
And is that because it's very similar in structure to a natural neurotransmitter like serotonin?
Yeah, so it's broken down by an enzyme called monoamine oxidase, and that's everywhere in the body. It's very high in the liver and in the brain, and basically DMT is gone in 5 minutes basically.
Mm-hmm.
So, but who knows? That's why science. This is why science is fun, because you can sort of go design these experiments and then see what happens. So it'll be interesting. Somebody will do the experiment at some time in the future, and then we'll know.
So how you probably have quite a large lab. What are the major projects or the major questions with respect to psychedelics that your lab is pursuing right now?
So we have basically two main questions that we're studying. The first is, so just to let your listeners know, I actually have the only grant to study LSD. So there's one person who has an NIH grant to study LSD, and I'm the only one.
In the US or in the world?
In the US. So the National Institute of Health only funds, there's literally only one grant to study LSD, and I have it. And I think there's only one other grant to study psychedelics, basically, and that's by Adam Halberstadt. For the last several decades, there have been very few labs that have studied psychedelics. So my lab has studied it, for many years, but mainly on the side because I never had funding to study psychedelics. It's only in the last 3 or 4 years that I was able to finally get a grant. And because of this, we actually don't know much about how psychedelics mediate their actions in the brain. And so a good chunk of our work is basically trying to understand, initially at the most fundamental level, how psychedelic drugs mediate their actions. And the idea is that once we can, since literally nothing is known about them, I mean, literally. And the idea that I've had is that once we can understand their actions at the most fundamental level, which is at the atomic and the molecular level, then we can use this as a toehold to basically go out at more sort of coarse-grained levels of understanding. So the single synapse, the neuron, and then the brain. And the inspiration for this sort of very reductionistic and sort of molecular approach to psychedelic drug action is inspired. I was inspired when I was a student by, Watson and reading about Watson and Crick, where they, basically solved the structure of DNA. And when they solved the structure, then this basically opened up a universe of knowledge. And, basically everything that we know about DNA, RNA, molecular biology, springs initially from the discovery of the structure of DNA. So the Moderna vaccine and the Pfizer vaccine that everybody is going to take probably soon is a nucleic acid based vaccine. It's an mRNA vaccine. And basically you can draw a timeline between the development of the vaccine and the discovery of the structure of LSD. Okay, is that clear at all?
Mm-hmm.
That's the idea. If we can understand initially at the most fundamental level how these drugs mediate their actions, then that will ultimately allow us to deconstruct or reconstruct the entire activity of the drugs all the way up to the level of the human. Okay.
Yeah, that makes sense. If you understand the chemistry and the biochemistry at a basic level, you understand exactly how each of these drugs are sitting in receptors, how they're doing that differently in different spots. That sort of unlocks the door to all of the other big questions that one might ask.
Right. So I call this the holy grail of psychedelic drug action. Okay, that's the first. We have to understand that first step. So I would say the last nearly 30 years have been devoted of my work in my lab have been devoted on and off to ultimately understanding that first step. We finally understand it. So a good chunk of my lab is devoted to that, and then we're sort of heavily invested in using that information to create new medications. So we're basically jumping from the most fundamental insights directly into medication development. So the idea is now we know how psychedelic drugs interact with the receptor. Can we use that information to design drugs that maybe are better than drugs like psilocybin for treating depression, anxiety, and so on, without perhaps without the psychedelic effects. We'll see. Whether that's possible or not, I don't know. But we're actually heavily invested in testing. I would say testing that hypothesis.
So the idea that a lot of people have right now is for a drug like psilocybin, it's showing promise for treating certain things like depression, say, or end of life anxiety. But of course, you're going to trip on it. So you're going to administer this to a patient, or they have administered this to patients under supervision. It's going to take a number of hours for you to have this experience. And that's obviously very time consuming. And it would be nice, in theory, if you could design a drug similar to psilocybin or something that had the therapeutic benefits that clinicians want, but doesn't require a 6-hour hallucinatory experience. So you're trying to design drugs that do one thing without the other things. How likely do you think that is? Some people think that we definitely will be able to dissociate those two things. And I know that other people think that the classical psychedelic effects might actually be an integral part of the therapeutic benefits. Do you have a perspective on how likely it is that we can dissociate those things?
I have no opinion.
Gotcha. So it could be anything.
Completely agnostic, yeah. But we, thanks to the DARPA, which is the Defense Advanced Research Projects Agency, they've given us a hefty award to basically test that hypothesis. So it's a hypothesis, and we have some unpublished data that suggests that which is, I would say, suggestive that hypothesis could be true, but ultimately we're never going to know until we get the right compounds and get them into humans. And we'll know right away, basically, right?
Yeah.
So in the very first clinical studies, we'll know if the drugs are psychedelic or not.
It's one advantage to humans, I guess, over mice is you can ask them.
They'll tell you right away. Yes. Now, I just want to talk a little bit about psilocybin and its potential for treating depression and other things. And I don't know if this is what you wanted me to talk about, but I just want to say a few things to listeners here about this. So, as mentioned, I spent many, many years as a psychiatrist and actually helped design and execute clinical trials for many years when I was at Case Western Reserve University, and I've consulted with pharmaceutical companies and started companies and so on. So I have a lot of experience in this space. And when I saw the initial phase 2 clinical data with psilocybin, my mind was completely blown because the effect size was so huge.
And describe the effect for us. What exactly did they find?
So they found that after a single dose of psilocybin, there was a hugely beneficial effect on depression. And these were in patients that were quote unquote treatment resistant. These were people with severe depression, for which standard treatments didn't work. Initially, they were people with cancer who were facing end of life issues and had anxieties related to this. And remarkably, after basically a single dose, now they actually gave fairly large doses to these individuals under very carefully controlled conditions. There was this really dramatically beneficial effect on depression and anxiety, which was maintained. And the longest follow-up now is 6 months, and a significant chunk of the patients are still symptom-free after a single dose. So this is truly remarkable. And I was asked to write a little commentary on at least one of those, on 2 of those studies, which I did. So it's pretty amazing. Now, and of course, there are anecdotal reports as well. But based on this really dramatic response, the FDA, the U.S. Food and Drug Administration, has granted breakthrough status to psilocybin for at least 2 different indications. So this is pretty amazing, and they're both in what are called phase 3 clinical trials. These are what are called registration trials. These are the studies that have to be done with large numbers of patients to demonstrate the drug is safe and effective before it would be approved for use by the FDA. And I anticipate that my prediction is that it will again show a big effect. Mm-hmm. So that's the positive. Now, what's the negative? Well, when you drill down into the clinical trial design, which I have done, one of the things that was pretty surprising to me is that when they recruited patients for the study, 90% of them were ultimately disqualified from entering the study. Mm. And they were disqualified for many, many reasons. One of which was inability to form a therapeutic, a psychotherapeutic interaction with a therapist. Okay.
Meaning they attempted to do therapy and it just wasn't working out?
Well, it's not entirely clear what the criteria were, but that's within the in the papers. The other is if there's a family history of serious mental illness. Mm-hmm. So if you have a family history of schizophrenia, so I have a family history of schizophrenia, you're not eligible to take the medication. Mm-hmm. Because there's a concern that in susceptible individuals, psychedelics could induce or exacerbate underlying psychotic conditions.
And is that a fear where we just want to be conservative in case that is true, or is there actually clear evidence that you can have a triggering of a psychotic episode from psychedelics?
So there are no controlled trials.
I see.
So we don't know. The other thing was that they excluded individuals that were suicidal. They also excluded individuals that had more than 3 prior psychedelic experiences, which I thought was interesting. Mm-hmm. That actually may exclude a lot of potential people that would be enthusiastic about taking psilocybin.
Mm-hmm. I mean, that one's sort of understandable to me because you might think, well, if it's people who are enthusiasts, that's another variable. These people might be more likely to report and display effects above and beyond what's actually going on.
At any rate, 90% were excluded.
So it's a question of generalizability. Is that what you're saying?
Yeah, and if you look at other recently approved sort of transformative treatments for depression, so there are 2 of these. One is ketamine, and the other is brexanolone. So ketamine is approved for treatment of depression, typically used for people that are suicidal, and has sort of a very rapid antidepressant effect, but it wanes usually in a couple of weeks. And if you go back to the clinical trial data, about 75% of the patients who were deemed eligible for the study.
Okay.
Which is more, I would say, more typical.
You would exclude a quarter rather than 90%.
Rather than 90%. And that's the case for the 2 big trials of psilocybin. So this is not just one trial. And the brexanolone is sort of an interesting comparator as well.
And what is that? What does that do?
It's technically what's called a steroid. So it's it's in the same chemical class as cholesterol or anabolic steroids or estrogen, testosterone. And it was approved for postpartum depression. So this is depression that women have after they give birth, which can be very serious. And it's given as an infusion over 60 hours. Okay, so pretty pretty noxious treatment, a continuous infusion for 60 hours, but it has this remarkable effect, in some ways similar to psilocybin. But less than 50% of the patients in those trials were excluded. Okay. So that's a little concerning to me. Mm-hmm. That's the downside. And it makes me, so the concern I have is that large numbers, so as I said, I expect psilocybin, will have this robust effect in the phase 3 clinical trials. If I was a betting man, I'd be willing to bet on that outcome. But it may come with a lot of strings attached in terms of patient eligibility, and so may not be, ultimately, will not be suitable for the general population. So I hope we can improve on that in some way, and that's a big sort of a big reason that we're spending so much time and effort in sort of formally testing this hypothesis that we can separate those two effects. If we can't, then hopefully we can come up with something that has a little better safety profile than LSD. There are some safety problems with those drugs, which I could go into, but basically preclude sort of microdosing and things like this for long-term treatment.
I do want to get into that because I have heard that for some of these, but I want to ask first, you've been studying psychedelics to some extent for decades, but it's only really in the past few years that's become this new sexy area. Right. You've been doing it for a long time. Where did this interest come from? Was it always there?
Yeah, ever since I was a teenager. So I grew up in Montana, and sort of the tail end of the 60s, I was a little kid, basically. And I knew about people who had taken LSD and actually had bad experiences. One person I knew ended up in a psych hospital for months. And as I said, there's this family history of schizophrenia. And when I was literally when I was 13 or 14, I started reading up about these things. In Montana, you don't have much to do. There's not a lot going on there. But we did have a public library, and I was taking high school chemistry, and so I realized these are drugs. This is a drug that's causing this effect. So drugs like LSD, clear to me as a 14-year-old, is affecting how people view reality. And on the other hand, there are these other drugs that people take who are psychotic, and when they take them, they're not so psychotic. And I just found this really interesting. What the hell is going on here? And so I was just interested in that. And it wasn't until I was in college, there was a visiting, basically this professor from Oregon came through one week to this. I was going to this small college in Montana, and I think he was probably coming to Montana to go fishing or hunting or something like that. But as an excuse, he gave a lecture to the biologists, and he was a neuroscientist, and he showed this diagram of synaptic transmission, and he talked about receptors. And that was the first time I'd ever heard the term receptor in my life.
And when was this?
This would have been in 1973 or 74, maybe 75, somewhere in that, maybe 1975.
Okay, mid 70s. You're learning about receptors for the first time.
The very first time I'd ever heard the term receptor. And I just said, wow, that's what I need. That's what I need to find out about is receptors.
An epiphany.
Yeah. It wasn't quite an epiphany, but it was like, what are receptors? This is really cool. And, there was no research, and I was very interested. I had this interest in drugs that basically drugs that make people crazy and drugs that make them sane. Okay. I had this interest in this. And I, since I was 14, I decided I was going to become a psychiatrist. I didn't know how that would ever occur, but that's what I was going to do. And so I got into medical school, and there was a guy there that studied receptors. And I asked if I could get in his lab, and he said yes. And so I got in, I started basically volunteering in his lab, and then got a PhD on opiate receptors, basically. Even though my interest was, basically psychedelics and antipsychotic drugs, that was my interest, were those two things. He didn't study them, so I studied opiate receptors. And then when I finished my PhD, I went to the National Institute of Mental Health to do a postdoc, and that's when I started studying, what I study now, basically. The psychedelic receptor. It's also the receptor for one of the receptors for what are called atypical antipsychotic drugs. And I must say that when I first started studying this receptor, I didn't start studying it because it was the receptor for LSD or for antipsychotic drugs. It was because nobody in the lab was working on it. And the guy I was working for was a really famous neuropharmacologist. And he just said, Bryan, you should study this receptor.
And was that the kappa opioid receptor?
No, that was the 5-HT2A receptor.
Oh, okay.
So, yeah.
Interesting.
So I got my PhD on opiate receptors. And when I finished my PhD, I vowed never to study opiate receptors ever again in my life.
Why was that?
Because at the time, the field was filled with all these really interesting characters. Most of whom are sadly dead now. But you would go to these scientific meetings and everybody would be yelling and arguing with each other. And I just decided I didn't want to be in a field with these crazy people who didn't get along with each other.
Okay.
So when I went to the NIMH, here was this serotonin receptor nobody knew anything about, the 5-HT2 or the 2A receptor, and the guy I was working for said, Bryan, you should study this. So I did, and then lo and behold, it turns out to be the receptor for LSD.
So why did he think you should study it if that wasn't even known yet?
So I don't know if this is going to be interesting at all, but it was for a crazy reason. Okay. So it turns out that serotonin doesn't bind to the 2A receptor very well. Technically it has what's called low affinity. And there were some unusual things about the way the receptor was regulated, which I won't go into for this audience. It's sort of technical. But this guy I was working with, his name is Erminio Costa. He sadly is dead now. But at the time, he was literally the most famous neuropharmacologist in the world. He was a member of the National Academy of Sciences. The lab had 50 postdocs in it.
Oh, wow.
It had 2 floors at the NIMH. and was an amazing place to work. So he actually thought that there was another neurotransmitter, perhaps a neuropeptide, that bound to the 5-HT2A receptor, not serotonin. Okay.
Mm-hmm.
And so he said, Bryan, I want you to find the peptide that binds to this receptor. Okay. And he was a really smart guy. So this is around the time that enkephalins were discovered, the endogenous ligands for the opiate receptor, endorphins, dynorphin for the kappa receptor, and so on. So it wasn't that wild of an idea to suspect that this could be another receptor for something else in the brain that we don't know. And so I spent about a year or a year and a half in the cold room trying to isolate the peptide for this receptor. And I went to him and I told him, it's serotonin. I'm sorry. It's a serotonin receptor. He almost kicked me out of the lab. He was very angry.
Anyways, you kind of happened to run into this receptor, and then it turns out to be the quote unquote psychedelic receptor.
Yeah.
And I guess the rest is history.
Yeah. I just stuck with it.
Wow. Wow. And so did you always
no insight, no particular insight on my part.
And so you had this interest in psychotics and antipsychotics. You had this interest in receptors and psychedelics. You had this family history in schizophrenia. Did you stay away from ever experimenting yourself with psychedelics for that reason?
Yeah, it's if you have a family history of schizophrenia, I don't think anybody would recommend that you take psychedelics. So.
Mm-hmm. Interesting. So what do you make of today? The last 4, 5, 6 years, you now have this renaissance or boom in research on psychedelics. You also have a lot of money going into the private sector for startups to develop.
A huge amount of money going into the private sector. Yes. Yeah.
What do you make of this whole phenomenon? Is it mostly exciting? Are there any cautionary notes you have for people? Are things already getting overblown at all? What's your general sense of the research that's going on?
There isn't. So there is. I'd say the clinical trial research from the UK group and the Hopkins group and the Vollenweider group. So those are sort of the Switzerland group. Mm-hmm. They're doing, pretty good, pretty excellent human research.
So at Hopkins, that's the people that many listeners may have heard about in the news in the US right. that are working on psilocybin. And in the right. UK, is that Compass Pathways?
No, it's Cathcart Harris and
okay.
And Nutt.
David Nutt.
David Nutt. Yeah. And then in Switzerland is Franz Vollenweider. Of course, Franz Vollenweider, like me, has been studying, psychedelics for decades. Mm-hmm. For decades, he was the only person doing human studies with psychedelics. So there is this handful of clinical groups that are doing, really, really excellent clinical studies. Finally, controlled clinical trials. Mm-hmm. Finally, in terms of basic science, there isn't a lot of really good basic science still. So as I said, I literally have the only grant to study psychedelics from a basic perspective. And then there is this group, Adam Halberstadt and Mark Geyer. So Mark Geyer, I don't know if he's semi-retired or if his lab is not entirely active, it may still be, but he sort of has studied psychoactive drugs, including psychedelics for decades as well. They sort of do behavioral pharmacology, excellent behavioral pharmacology. And that's about it. I mean, there are some other people. Charles Nichols does some research, sort of on a smaller scale, but there isn't a lot of basic research that's being done. There are a number of, I guess David Olson does some as well, not directly with psychedelics, but sort of psychedelic inspired research.
That was the recent paper about ibogaine, right?
Ibogaine, an ibogaine derivative, yeah.
Mm-hmm.
There, but there are many companies, mainly looking at, seems to be mainly looking at psilocybin. Some of them have licensed. I think David Olson has a company. Charles Nichols has a company. They may have some new chemical matter. But there isn't much known about these compounds, these newer compounds. So we have this paradoxical thing that venture capitalists and rich people, sort of rich hippies, are pumping lots of money into psychedelic space, but there's not a firm foundation of science that underlies a lot of what's going on. So there's a lot of enthusiasm, but it's fascinating to me. We don't really know much about how these drugs exert their actions. Mm-hmm. And I don't know, if I was an investor, I would want to know a little bit more about that.
What do you make of the research? So you sort of study things at the basic level, the very small level, so receptors and chemicals and molecules. And then there's people like the group in the UK and elsewhere that are doing fMRI, brain imaging studies. Right. There's some EEG work out there looking at the global brain states and patterns of brain activity when people ingest psilocybin. Yeah. Do you follow that literature closely? And what do you think, what are the basic takeaways that we know about so far about how psychedelics are affecting global patterns of brain activity?
So I think it's mainly phenomenological data right now. I don't read their, I must admit, I don't read their papers that closely because I just don't, it's just at too fuzzy of a level for me. We are, I have a big lab, and so we are actually looking at things at a more integrative level. And so for the DARPA grant, we actually have a huge behavioral effort, mouse behavioral effort. And we have collaborations with basically the best neuroscientists in the world to look at neuronal firing and things like this. So for the DARPA grant, I've been able to bring together a team which in their respective areas, I think is literally world class. So we have one of the world's experts on what's called cryo-electron microscopy, this guy Yorgos Skiniotis at Stanford. The group that's basically the leading group in the world in sort of computational discovery of drugs, which is the Shoiket and Irwin group at UCSF. Basically the world's greatest person in terms of understanding the dynamics of receptor activation. This is Ron Dror at Stanford. The Duke Behavioral Core is running, Duke University Behavioral Core is running all of the behavioral studies at a level that really no one else in the world can do. And then we have a huge medicinal chemistry effort. So we have 5 full-time chemists. And we also have basically one of the leading people who does proteomics, this guy, Nevin Krogan, His lab at UCSF is trying to understand sort of the proteomic networks. We're doing transcriptomics networks. So it's sort of it's a Manhattan style effort that it's like a Manhattan project for understanding psychedelic drugs.
So you've got multiple teams, and you guys are looking at psychedelics at multiple levels, meaning you're zooming in very close to look at very zoomed in pictures of things happening at synapses.
The atomic level,
yeah,
the molecular level, the cellular level, the neuronal level, and the behavioral level.
Mm-hmm.
So, yeah, we have a huge effort in making, engineered mice for various things. And then we were basically combining this with drug discovery. So the goal is basically to deliver new medications. So it's unprecedented, basically. Mm-hmm. It's the grant is $27 million. So this is like for this sort of effort, this is a huge amount of money. And on top of that, I have this merit grant from National Institute of Drug Abuse, basically to understand at the molecular level what's going on with these drugs. So we have this. We're very fortunate to have this really, outstanding team of scientists that can bring sort of world-class brains to really a complex problem. Which is unprecedented, I would say, in the history of study of psychedelics. So our hope is at the end of the 4 to 6 year period that we'll not only have new medications, but we'll actually have some knowledge about how these drugs exert their actions at the brain.
Interesting.
Finally, basically.
So we could probably expect to see a body of work here, multiple papers.
Yes,
multiple
papers.
Which compounds are you focused just on LSD? Are you doing multiple different studies on different compounds?
So it's a big project. So we're in the process now. So there are 30 investigators working on this. So 30 scientists just on this project, and then with my other funding, there are probably another 4 or 5, so maybe 35 people working on this project. And so, in terms of the basic biology of psychedelics, we're actually looking at hundreds of known psychedelic drugs. And we're in the process of putting together basically a paper that will serve as the database for our understanding of psychedelics. So every psychedelic drug that is known to be psychedelic, we're looking at right now. And at many different levels. Mm-hmm. So the receptor pharmacology, the signaling, transcriptomics, proteomics, and so on.
So you're building a database essentially for researchers where every known psychedelic will have a complete, or at least a very thorough database of which receptors they bind to, what they do at those receptors, how genes are turned on or turned off because of that. Right. And all of that will be available for researchers to tap into.
Yep. It'll be free online.
For anyone.
For anyone.
That's awesome.
Yep. And we're also, so that's just a small part of this project. Mm-hmm. That's like this much of that project. Mm-hmm. Okay. The big part of the project is actually to come up with what's called new chemical matter. So what we want to do is we want to come up with completely new structures of compounds that will activate the receptor in specific ways. And so we know that the signaling of these receptors is extraordinarily complex. And we're basically we're designing and discovering completely new chemical entities that look like nothing that has ever been seen on the planet Earth, to activate these receptors in distinct ways.
So literally drugs that don't exist on Earth in normal biology, and therefore drugs that have the potential to activate receptors and have effects that we've never seen before.
Right. And of those, we're basically doing chemical optimization to make them have better, what are called drug-like properties or medication-like properties.
Mm-hmm.
And we're optimizing them ultimately to be candidates for testing in humans.
And so when you optimize something for candidate to be tested in humans, what exactly does that mean? You're minimizing the risk of side effects and things like that?
Oh, it's a huge amount of work. So it's, you have to, so there are what are called drug-like properties. So these are things like, can you take the drug by them as a pill form, and will it get to the brain? Okay. So that's one. So that process is very complicated.
Mm-hmm.
There are many things that regulate that process, and all of those things need to be optimized and characterized. And then you want to make sure that it doesn't have, the potential for serious side effects. You want to make sure it's specific for the receptor. And you don't want it, you wouldn't want it to disappear within a minute of entering the bloodstream, right?
Mm-hmm.
And you probably wouldn't want it to hang around for days. So you want to optimize that as well. So there are just many things that need to be taken care of. A lot of these can now be predicted with computational tools, but ultimately have to be tested and quantified. So there's a huge science involved in this, but that's one of the things. So to give you a sense, so we're about 6 or 7 months into the project. And already we've evaluated around 500 novel compounds.
Wow.
That have been created just for this project.
Oh, wow.
Yeah. And we've already put 2 of them in mice.
Wow. So it's going to be this, enormous body of work with this really rich database that anyone can tap into. And like not only drugs that we know about, but essentially alien drugs that you've made from scratch.
Right. Yeah. This is cool.
Yeah.
Yeah.
So
thanks to your tax dollars.
Literally, yeah. So how does like someone like DARPA decide that this is where they want to fund research? Can you give us some insight into how research funding actually works and where they decide to point things?
You would have to talk to them, but basically they put out what's called an RFP, request for proposals. And what they wanted was the title of the thing was Biased Agonists as Rapidly Acting Neuropsychiatric Agents. It's sort of a mouthful. Focus Pharma, I think, was the name of it. But the idea was basically what I outlined to you. Can you come up with a non-psychedelic psychedelic?
Mm-hmm.
So is it possible to create a drug which is effective like psilocybin, but is not psychedelic? So the idea is, can you come up with a drug that you take 1 dose and you're cured?
Yeah, that would be nice.
Wouldn't that be great? Yeah. So DARPA I don't know how they come up with these ideas, but DARPA likes to, my understanding, I can't speak for DARPA, but from what I understand and talking with other people is they like to pick impossible projects. And
so it's like the moonshot effort at Google or something.
So they know that many of them will fail, but if the upside is huge.
High risk, high reward.
Right.
Yeah.
So the idea is, it'd be great if we could, come up with something where if you're depressed, you take a pill and the next day you're not depressed. Wouldn't that be wonderful? Right.
Mm-hmm.
that would transform humanity. So that's the task they gave us. And they gave us a certain time schedule.
What was the time schedule? Yeah, what's their expectation here?
4 years.
4 years. So they give you $27 million, but there are strings attached.
Oh yes.
And so what would they expect you to have to show for yourself after 4 years? Is it a certain number of papers? Is it a certain kind of result?
They want two compounds basically ready to go.
What happens, just out of curiosity, what happens if you don't come up with two?
Well, so there are milestones that we have to meet along the way. And if we don't meet our milestones.
I see. So it's phased funding.
Yes. Yeah.
I see.
Yeah. So there's a very high level of supervision and oversight. Mm-hmm. So we meet very frequently with the program managers and, give updates and all this stuff. It's nothing for people that have NIH grants. It's nothing like it. The level of oversight and expectation is extremely high. It's more like if you were in a biotech company. Mm-hmm. It's that sort of pressure. You have a certain timeline. You have things that need to get done. If you don't get those things done, or if it looks like your project is not going to work, they want to find out as soon as possible so they can cut the funding.
I see.
So it's a pretty tight it's a pretty tight schedule. It's a very difficult project, but we have an awesome team and we're making great progress. So I think we'll get to the finish line. But we'll see.
Yeah, no, that's amazing. I mean, it sounds like a heroic effort. It's a distributed team. And I mean, I'm really excited to see what comes out of that. I mean, I love the fact that you guys are going to have this database. I used to work in academia. I now work in the private sector. So I can no longer just put all of the data I have access to out into the world. And so I appreciate just hearing that things like that are going to happen.
Yeah.
The other thing that I wanted to talk to you about, because I've heard this about you. In fact, the first time I heard it about you was, I've never met you for the listener, so I've never talked to Bryan before, but I was in the same room as you one time, and it was, I don't know, 6 or 7 years ago when I was a graduate student at Harvard. You gave a talk, and then there's often a student lunch after a talk. So a scientist will come in to give a talk, present the research, and then
I think I talked about the structure of LSD at that talk, right?
No, this was about dreads, I think.
Oh, dreads. Okay.
Yeah, this is actually my introduction to Bryan Roth. Was when I started graduate school, I started using these things called dreads.
Oh, okay.
Which are tools. They're GPCRs, receptors, like we were talking about. They've been modified.
Right.
And you can use a bag of tricks in the lab to put these receptors into animals. And it allows you to then activate those receptors, say, and manipulate the animal's behavior to see how certain neural circuits are working. So I was using dreads to make animals hungry. I was working on AGRP neurons.
Okay. Oh, yeah.
So you can put these things into animals, make them do something, in this case, eat. And so I learned about Bryan Roth. So I went to see Bryan Roth speak. You talked about dreads. And then in the student lunch afterwards, where there's sort of a Q&A with the graduate students, you mentioned that you were a Zen meditation practitioner.
Right.
If memory serves me correct, I think you said that you meditated for something like 4 hours every morning.
Yeah.
So walk us through that. How is that your schedule? Is it 4 hours every day? And
yeah, at least 4 hours a day.
Is that continuous all at once?
Let me just, before I get into that, let me just say that one thing you may not know about dread technology is that I invented it to deconstruct psychedelic drug action.
I did not know that. Yeah.
So that was the reason I invented it, basically. And the idea was we would make, a GQ, what we now call the GQ dread. We would put it into layer 5 pyramidal neurons, turn it on, and recapitulate the actions of LSD. So that was the idea, basically.
Okay, so I think what So what you just said was you were trying to create a receptor such that you could put it into particular neurons in the brain, turn it
Right.
on without a drug,
Right.
and make the animal essentially behave as if it had taken LSD.
Right. Yeah.
Wow.
Yeah.
And it turned out just to be this great tool that allows neuroscientists to selectively turn on or turn off neurons in order to figure out how the brain is actually doing what it does.
Yeah. Yeah. And thousands of labs around the world use it. It's quite amazing. Sadly, my lab doesn't use the technology. But
anyways,
yeah. With that segue, we can talk about
from dreads to meditation.
From dreads to meditation, right?
So I remember hearing that. This is several
Right.
years ago, and I was like, "Did he just say 4 hours a day?" So, is that all at once, or is it broken up?
So I usually get up around 3 or 4 in the morning, and I will. I have actually a little meditation room that I set up here, and at home, and of course everybody is asleep. My wife is asleep. My dogs are asleep, and I meditate until around 8 o'clock, basically.
Wow.
And then I may meditate in the afternoon or at night, but that's my typical day.
And how long do you sleep each night?
Usually go to sleep between 8 and 9, so I get plenty of sleep.
Okay, so it doesn't affect your sleep drive.
Well, so I get like I don't know, 6 hours of sleep a night, probably.
Okay, so that's probably the low end of the normal spectrum.
Yeah. I get by with a little less sleep probably than the normal person, the quote unquote normal person, typical person. Yeah.
And what style, is there a particular type of meditation you're doing?
Yeah, I practice Zen. And
what is Zen as opposed to mindfulness meditation or some other form of meditation?
So there are basically three, I guess you could say three types of Zen meditation. And I'll just discuss all three, and then I'll tell you what I do. So sort of the one standard way of Zen meditation is what's called following the breath or counting the breath. Basically, when you breathe in, you count 1. When you breathe out, you count, you're not doing this when you're talking, but basically in your mind, you're breathing in 1, and then 2, 3, 4, all the way up to 10, basically. And when you get to 10, you start over again. So that's called counting the breath. And that typically is refined to following the breath. So you follow the breath going in, and then you follow the breath going out. Or it could be you only count the exhalations. So those are variations. So that basically sort of basic breath practice. So that's a very common type of meditation. It's called breath practice. And typically when people start out with Zen, practicing Zen, they start out with breath practice. And you can stay with breath practice your entire life. So following the breath is a complete practice of meditation. Once one gains some, I would say, facility or is acquainted with breath practice for a time, so that practically you wouldn't, if you were counting your breath, you wouldn't lose the count for 15 minutes or so. So the instruction with breath practice is, if you forget what number you're on, you go back to 1, basically. So if you're able to do this for 15 minutes or 30 minutes mainly without losing the count, then if you wish, you can go to another practice. And there are basically 2 other practices in Zen. The first is what's called koan practice, Koan. And the other practice is what's called, they have these Japanese terms, shikantaza. And koan practice is so let me back up. So breath practice you can sort of do on your own. You could do it on your own without a teacher. It's helpful to have a guide, but I've just now taught you the basics of breath practice. You could go home and practice basic Zen meditation with that practice. Koan practice or shikantaza, it definitely is helpful to have a teacher. So koan practice is typically only started during a Zen retreat of at least typically of 7 days. So you wouldn't ordinarily take up koan practice if you didn't have, I won't say if you're not dedicated sufficiently, but if you don't have the time to spend a week meditating, where you're, and in a Zen center, this would be you get up at 4 a.m. and you go to sleep, lights are, you can go to sleep around 10 p.m. And during that time period from 4 a.m. to 10 p.m., you're meditating most of that time. Okay. In a meditation hall with other Zen practitioners. So koan practice is focusing your attention on a question. And a typical question, I think a lot of people have heard of this one. What is the sound of a single hand? Or what is the sound of one hand clapping? But really it's what is the sound of one hand? Would be a koan. That's a koan. Another koan would be show me the face before your parents were born. Show me your face before your parents were born. That might be another koan. Okay. But typically the first koan that people work on is a koan called mu, M-U, or some people will say the koan is the word no, N-O. And it's a very short koan where a student comes to a Zen master and he asks the question, does even a dog have Buddha nature? And the Zen teacher says, no, or mu in Japanese. And that's the koan. And then you focus on that word, mu. And so that's koan practice. And, it sounds weird, right? Mm-hmm. Kind of strange, focusing on this word mu. What is this word mu? What does this mean? Mu. So it's very helpful to have a teacher to at least understand what the practice is. And so
is there an answer to that, or is it meant to be unresolvable?
So there's a resolution of the koan. And in koan practice, the goal, if you could say is a goal, is to see things as they are, at least initially with a sudden insight, what's called kensho. And in common parlance, this would be having an enlightenment experience. Okay. So the goal of koan practice is to have that experience. We would say in Zen to have the same experience that the mythical Buddha had, at least to a small degree, some tiny degree, to see things as they are, to see into the nature of reality suddenly. And then the goal of subsequent koan work would be to clarify that vision, clarify that understanding, so that that understanding is permeates your life, basically, so that you are the understanding. And that's why it's, very helpful to have a teacher. It's nearly impossible. It's not entirely impossible, but it's nearly impossible to do it without to do that, to have that sort of practice without a teacher. For most people, a teacher is essential.
So I take it that you did have a teacher at least at one point.
Well, I have a teacher, yeah. So I've been practicing Zen for 30 years with the same teacher. And then the third type of Zen practice is what's called shikantaza, which is probably the most in some ways it's the most difficult of all practices, or the easiest of all practices. And shikantaza is, you could say, I'll just use some sort of jargon here. It's being in it would be non-dual awareness.
Mm-hmm.
Are you acquainted with the idea of non-dual awareness?
I am, but can you describe that?
So that would be where there's no sense of a separation. So this is sort of what people describe it as a peak psychedelic experience, right?
Yeah.
Non-dual, I and the universe are one,
mm-hmm.
right? So a kensho, is that experience.
Mm-hmm.
I and the universe are one. Above the heavens and below the heavens, I am the honored one. I walk the universe alone. I am the universe, okay? That's kensho. That's also the peak psychedelic experience, from what I understand, at least from what I can read. So shikantaza is that sitting.
And so I take it that you've experienced that state, and if so, is that something that has happened multiple times briefly, or are you able to sit with that type of non-dual awareness for extended periods of time?
So we don't talk about what our own experience is in Zen. There's this little, but I can speak about it in the third person.
Mm-hmm.
I think that would be fair. There's this funny cartoon I just saw that I sent to a longtime friend of mine, which is sort of a takeoff of Fight Club. So the movie Fight Club.
Mm-hmm.
So it says, it's, who was it, Brad Pitt, and who was the other guy in Fight Club?
Edward Norton.
Edward Norton, are looking at each other, and one of them says, the first rule of Satori Club is we don't talk about Satori.
Okay.
So, shikantaza is basically a moment-to-moment practice. So it would be, it would be every moment of awareness is remembering or is recognizing the non-dual nature of reality. So that would be the practice of shikantaza, is instant by instant awareness of the non-dual nature of reality, if that makes any sense at all.
Well, I think it does, but I think it might not to most people. So
Yeah.
why don't we, so in the context of, say, like neuroscience speak,
Right.
would it be fair to say something like this, that your Zen practice or certain altered states that you can enter into through meditation or from other means?
So let me just back up here. So in Zen, one of the marvelous things about Zen is that these are not altered states. So this is the natural state of the mind. This is so Zen is part of Buddhism. I don't want to get too philosophical here, but in most schools of Buddhism, this is what's referred to as the natural condition.
Mm-hmm. So would that mean it's
not an altered state at all? The mind is clear.
It's something prior to all of the differentiated forms of awareness that one may have had through conditioning throughout life. We would say it's actually always there, but it's not recognized. Mm-hmm.
It's there all the time. It's just that
would you describe it as maybe perhaps the form of experience into which all of the particulars are put?
It's more like the mind is just distracted from recognizing things as they are.
Mm-hmm.
More like that. It's more like just distraction. And the various techniques of meditation are basically ways to remove the noise, this noise in the mind, this distraction, so that you can see things as they are. So it's not in any way an altered state. In that way, it's different from the psychedelic experience. So people that have a psychedelic experience, they would, have this experience of oneness. And it would remain sort of a pleasant memory, right? I had this experience of oneness. It was great. Where did it go?
Mm-hmm.
Right? I remember it. It was wonderful. Where is that now? So in Zen, it's sort of moment-to-moment recognition of just a recognition of this is the way things are, basically. Now, how can you say this in neurobiological terms? So one way I like to think about it is what's called the default. You know about the default mode? Okay.
Yes. Let's describe that for people, though. What is that?
So it's basically the way the brain works. I won't say the mind. I'll distinguish the mind from the brain. It's the way the brain works. So the brain basically has things that churn around, sort of seemingly under their own, without any external force. And a lot of this is experienced as chatter in the mind. So when you're sitting down, listening to music or something, you're not only listening to music, there are thoughts basically going through your head, right?
Mm-hmm.
This is the default, to a great extent, this is the default network. It's churning all the time. Part of the practice of what meditative practices do is and one of the reasons the default network continues is that our attention is shifted to it. Okay. So if we didn't reflexively shift our attention to the thoughts,
They would run out of steam.
They run out of steam, basically. Yeah. They don't
So
disappear entirely, but you then recognize this sort of silence between the thoughts.
Mhm.
That's sort of one way of looking at it. And the silence is the natural state of the mind, is this silence, this luminous, what one could call luminous awareness. Now that's not Kensho or Satori or enlightenment, but that to recognize that silence is an initial first step. So that's what happens basically. So that's why I say it's a natural. It's just things. It's the way the mind is naturally, but we just don't see it because we're distracted with basically the way that things work. Our mind works. It's continually generating this chatter in our heads, which we pay attention to.
And what so on the one hand, this makes sense, but I could see how someone would hear this and think, well, if this is the natural state, and if meditation is good for you, if it's something you should do because it's somehow better to recognize these things that you've just explained to us, why on earth would there be a default mode that has evolved in the brain that is chattering to begin with?
So I don't know, but it seems to be the way things are. So I don't profess to know anything about the way the mind is constructed, other than to say there must be some evolutionary advantage to having a default network. I don't know what that is.
So how did you get in? How long have you been practicing again? How long have you been doing this?
So I actually started meditating when I was probably 16 or so. And
what was the original impetus for you? Was it just curiosity?
Oh, no. So sort of I don't know if this is a funny story, but my dad heard about transcendental meditation as a way to keep your kids off drugs. And as I said, it was sort of the tail end of the 60s. And there was this young woman who had spent time with Maharishi Mahesh Yogi, who was the head of transcendental meditation and was a transcendental meditation teacher. And she had come back from wherever she had trained with him, India or whatever, and had set up shop as a meditation teacher. And she had a special deal for families. And so my dad had all of us take the transcendental meditation course. And so I started meditating, I think when I was 16 or 17, did transcendental meditation. And it was actually very helpful for me.
And what is TM? How does that work?
So it's what's called a mantra practice, where you, instead of focusing on the breath or a koan, you focus on a sound, basically. So this is not sort of a TM mantra, but one mantra might be the word Om. So you focus on the word Om. So they basically give you a word that you focus your attention on. And the practice is basically like every meditative practice. You focus on the word, and then when your mind wanders away from the word, you basically bring your attention back to the word. And the mind steadily gets quieter and quieter. And it was interesting when I took TM, they said the goal of TM was cosmic consciousness. They didn't explain what cosmic consciousness was, or even why that might be something you would want to have. But it was very helpful for me decreased my anxiety. I think I got along better with people and helped me focus. So I practiced TM for a number of years, and when I got to medical school, for various reasons, quit practicing TM. Then when I was finishing up my psychiatry training at Stanford, I took up Zen, and I've been basically doing it ever since.
And how did that get started? You mentioned having a teacher and needing a teacher. How does one actually go about finding someone to help with that?
Well, so I went to the Yellow Pages, and now
maybe we should explain the Yellow Pages.
These days you would just go to the network, go to the net, and go to Google Maps and type in meditation teacher, and most any place you are, you'll find some. But I was in Cleveland at the time, and I'd sort of begun teaching myself Zen. But I realized it'd probably be a good idea to learn about it in a more formal way. And I had this book, sort of a famous Zen book called The Three Pillars of Zen. And at the back it said, for further information, contact the Rochester Zen Center. So Rochester was, probably 200 miles away from Cleveland. So I called them up on the phone and I said, I want to learn about Zen. And they said, okay, why don't you come to an introductory workshop and we can teach you about Zen. So I went, basically. It cost, I think it would cost $75 for it was a 2-day workshop. You got free meals and free room and board, basically, for $75. So it was a good deal. That was actually a good sign. They didn't charge me $1,000 for 2 days, $75. And I met the teacher, and then started practicing, basically.
And so you said that the stated goal for TM was cosmic consciousness. You talked earlier about this form of non-dual awareness.
Right.
Is it accurate to say that what you're describing there is a form of conscious awareness in which your brain's construction of a self, of a model of itself, is somehow gone, but awareness is retained?
I wouldn't say that. I think what's commonly described in the literature is that one sees through the illusion of a separate self. So if you read accounts of people who quote unquote who are deeply enlightened, they would say that the illusion, so they recognize the illusion of the separate self, basically. That's what they would say. The illusion is still there, okay? And at the same time, the awareness of the non-dual nature of reality is there simultaneously, is probably what they would say.
Gotcha. So those are two different things. This recognition of the illusion of a self and this non-dual awareness, those are separate but related?
It's the same thing.
It's the same thing.
Yeah. Yeah. They would say it occurs simultaneously.
I see.
So they would say, so there's this sort of classic Zen saying, before enlightenment, mountains were mountains and rivers were rivers. When enlightened, mountains are no longer mountains, rivers are no longer rivers. And after enlightenment, mountains are mountains and rivers are rivers. That's what they would say. I don't know if that makes any sense at all.
I suspect it will be puzzling to a lot of people. So
what they would say is that things are exactly the same. Mm-hmm. Nothing changes. Just the how can I say it? Just the mistaken belief in separation is gone.
Mm-hmm. And how would having sort of gone through
that make any sense at all? It's probably just gobbledygook. I'm sorry.
Well, I think for a lot of people, where you start to get hung up is this sort of mistaken perception, as you described it. They've never experienced it.
Right.
So it's like a negative. It's like trying to imagine a negative when you've never actually experienced the absence of this thing that's always been there.
Yeah. Yeah. Although I would say you do all the time, and maybe just don't notice it. So, when you see a beautiful sunset or,
anytime that you're quote unquote lost in the moment or fully engaged. Yeah. You're not thinking, I'm sitting here, I'm doing this. You're making the jump shot in the championship game. You're fully engaged in the act.
Right. Or you're eating a marvelous nacho.
Yeah. The point being that you can achieve the same state that you might more readily associate with the high octane peak experience. You can also have that exact same type of framing for the mundane everyday experience of eating a nacho.
Right.
Do you ever miss a day? Do you ever, you're in the middle of the week, you've got a big DARPA meeting coming up. Do you ever miss your meditation session?
No, I don't. With a couple of exceptions, there have been times when, like I've been on a plane for 24 hours, literally. So I'll meditate in the seat, basically, of the plane. But no, I don't think there have been any days where I have completely missed meditation entirely. In 30 years, maybe once or twice, basically because I was awake all night and couldn't formally meditate the next day.
Wow.
It's I just naturally wake up at 3 or 4, sometimes earlier, sadly, and I have nothing better to do, so I meditate basically, rather than other things I could do at that time of night. So yeah, I don't. I would say, if nothing else, my practice has been regular. I can't say that it's been particularly productive, but at least I put in the hours, if nothing else. I'm not particularly good as a meditator, but I do practice.
What is that? Well, what exactly does that mean to not be good at it?
I'm just being funny. Yeah. I mean, anytime in meditation, anytime the attention wanders, which will, I guess, unless you were the Buddha himself or, some greatly enlightened being, some mythical enlightened being, your attention is eventually going to wander. And it's always, basically the act of meditation is really the simplest part of it is, oh, my mind wandered. Let's, just make that movement. It's that recognition. And that, that's a moment to moment. That's always something you're practicing, because the nature of the mind is to sort of wander off.
Interesting. Do you follow the literature at all on meditation, like the neuroscience of meditation?
I do not.
Interesting.
Yeah. I have people have asked me that before. I have no curiosity about it at all.
Interesting.
It seems to work for me. That's fine.
Interesting. Well, as we wind down, Bryan, maybe I'll just ask you one or two more questions.
Sure.
What are you most excited about the next 1, 2, 3 years in terms of questions that you think might get answered by the work in your lab?
So for better or worse, I don't think that way. I've always had this deep interest in psychedelic drug action. But what gets me most excited in the lab is basically the latest data, which is unexpected. So I have a big lab, and people are working hard, and almost every lab meeting somebody has something which I get excited about that wow, that's cool. So
you're just always looking forward to the next surprise.
Yeah. 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 I mean, we've had some pretty intriguing thing. Sadly, I can't talk about it because we haven't written it up yet, but I have this amazing group of postdocs and students and professional people in the lab. And every week there's just something that's just really amazing that it's like, wow, I never would have guessed that. So I'm just, I don't wait for it, and I don't really think about it, but that's what gets me excited, is like new data.
Well, Bryan, thank you for taking the time. I really appreciate it. We just talked for almost 2 hours.
Okay. And I got another meeting here.
And we covered a lot. So thanks again, Bryan Roth from the University of North Carolina. Any final comments you want to share?
No. I always like to thank the funding agencies, the NIH and DARPA, your tax dollars at work. I'd like to thank everybody for that. It really is, we can't do it without government support. It's so essential. And, there are many things about our society which, sadly we that are problematic, but one of the really wonderful things about the society as it is today is that they support basic science. And, without that, we wouldn't have things like the vaccine for COVID-19, and hopefully we'll have better medications here before too long. So I just like to thank everybody for that. And thank you for your time as well. So.
Thanks, Bryan.
Okay, bye.